All right, so now up to another adventure. I want to compare two sequences that are similar, and I want to see how similar they are or where their alignment is. So from one of the last exercises, we had the sequence, a DNA sequence that was isolated from a bacterium that lives in the gut of an insect, and we had a short sequence there. It was only a partial sequence, and we didn't know whether it was messenger RNA or ribosomal RNA. But I want to compare, I think, first of all, we figured out that it was 16S RNA, and we can do that again quickly. But I want to find a way where I can actually align the two sequences and compare the sequences and see where the similarities are. So let's go to BLAST again. And firstly, I just want to show you again that the sequence we had from our activity, I didn't say whether it was mRNA or rRNA, so we keep the standard database and we do the BLAST database. We BLAST our sequence and let's see what we got. It should be Borrelia burgdorferi again. Okay, so this is confirming, just by blasting the sequence we had, is confirming that we have the 16S ribosomal RNA gene. But I want to now go and find a sequence at the NCBI. So I'm gonna go to genes and expression, and I will finding the sequence by looking for nucleotide database. I'm looking for Borrelia burgdorferi 16S, and I'm finding the actual submitted sequence, and I want to compare that sequence with the sequence I have. So I go to FASTA and I'm copying the sequence, and if you're looking at the sequence, actually pay attention. So what are the nucleotides for the DNA? So it's ATCG, and you can see throughout here there's a couple of N's, and that is usually a sign that there was a problem with sequencing. So again, just pointing that out. Always keep an eye open for something that doesn't look quite right, because ATCN is not really one of the sequences. But anyway, let's just compare the two strains, and let's compare the two sequences. And again, how do we do that? So we are going to BLAST, NCBI, analyze. We're gonna go to BLAST tool, but we're not looking for one sequence, what it means. So let's go and search for our standalone blasts, and I want to do the global alignment because I want to compare two sequences across their span. And so we have two entrance boxes here. So this is the sequence I just retrieved from the NCBI database, and now I'm looking for my sequence what I had in my activity before, and so we put them both in there, and let's align them and see. Again, just by looking at it, it's really hard to figure out where the alignment is, but this BLAST analysis will clearly help us to find that out. Okay, so now we have our sequences aligned, and let's just go back on the analysis. Here the descriptions. So the score is 13% identity, but again, this is the low number here is because one sequence was very short, and you can see that if we look at the actual alignment, is that the query was the whole 16S sequence, and the little sequence what we had was just a fraction of it. But you can see that it was, except this one nucleotide here where it was an N, it was basically 100% alignment. So again, that's another tool where you can align two different sequences and compare how similar they are, what area they overlap, or you can ask many other questions as well. So in this case, we aligned the small part of the 16S sequence of Borrelia burgdorferi with the whole 16S sequence. and we found that there was basically 100% overlap alignment between these two sequences. All right, yet another cool tool conquered. Happy bioinformatics skills.